Hi, I am Mrs. Sloan, and this video is for my honors biology students. This is part 2 of our chapter on cell division. Part 1, we focused on mitosis, and here in part 2, we're going to focus on meiosis. So I'm going to make myself a little bit smaller here and get us in presentation mode. Hopefully my dog will not bark, but you never know. All right. Okay, so in the descriptor of this video below are the notes that I give to my students. Column 1 is a scaffolding of the notes, and then I'll help you fill those out. Column 2, put in pictures and images that help you. All right, so on our notes, we are on chapter 5, and we are on section 5.3. Sorry, I can hear my dog. 5.3, reducing the chromosome number for reproduction, meiosis. So let's get started on that. So before we get too far, I want to remind you of what we learned in mitosis. We learned about the stages, right? Prophase, metaphase, anaphase, and telophase. We're going to see those same names come up again with the same key events that occur in each one of them. Remember, prophase is always when chromosomes become visible. Metaphase, you have chromosomes meeting in the middle. Anaphase, you're separating something. In the case of mitosis, you're separating sister chromatids. And then telophase, you're reforming the nuclear envelope. Overlapping, and mostly in telophase, is cytokinesis, and that is division of the cytoplasm. So that's what we did. You get clones of the original parent cell. If you have 1, 2, 3, 4 chromosomes in this original parent cell, each daughter cell is going to have 4 chromosomes. It keeps it exactly the same without any variation. So now we're going to change gears. In meiosis, what we're going to do here is we're going to take a diploid cell and we're going to generate haploid cells from that. Why? Because you're getting ready for sexual reproduction. And so what we want to do is have a union of sperm and egg to form a diploid zygote, that baby. So the sperm is haploid, the egg is haploid, haploid plus haploid will be a diploid baby. Now, when that baby grows up, it will do that by mitosis. Remember, my arms, my legs, mitosis. And later in development, it will start to do meiosis, and then be able to form its own sperm or eggs. So we want to remind ourselves what homologous pairs are. Homo means same, so a homologous pair of chromosomes means it has the same type of information. It may say 2 different forms of that information. For example, from your father or paternal side, it might tell you to have brown eyes, and your mother's, there could be a gene on here on the maternal side saying blue eyes, but they're both talking about eye color. Okay, so they are this homologous pairs are the same size, same shape, and code for the same characteristics, though alleles are variations on those traits. And I also want to remind you of what sister chromatids are, right? When this chromosome right here replicates, and what stage of the cell cycle does it replicate? Right? The S stage of interphase is when you form these sister chromatids. So this is considered one chromosome consisting of 2 chromatids. All right, so let's go into our notes on 5.3 overview. DNA replicates as before, forming those sister chromatids. You can see them right here, forming the sister chromatids. Homologous pairs. Cells initially undergoing meiosis are diploid. You have 2 copies of each type of chromosome, 1 from each parent. Okay, so this could be homologous pair number 7. And so you have dad's version of 7 and mom's version of 7. Remember, when you were formed, dad gave you a copy and mom gave you a copy. We have 23 homologous pairs, which means 23 homologous pairs, 23 times 2, 46 total chromosomes, 23 coming from our father, 23 coming from our mother. Okay, so we have 2 copies of each type of chromosome, 1 from each parent. Then if you recall, let me get to, here we go. Here's a homologous pair of chromosomes. This one codes for purple flowers, this particular version of coloring. This one codes for white flowers. And so this is a homologous pair. During the S stage of interphase, each of those chromosomes is going to replicate itself. And so alleles are alternative forms of a gene. Also, this one, they're both coding for color on the flower, one white, one purple. So an alleles are alternate forms of a gene found on chromosomes, alternate forms of a gene found on chromosomes. Mother's chromosome could say blue eyes, father's brown, etc. Okay, now when you do meiosis, in essence, what you're going to do meiosis 1 and meiosis 2. Meiosis 1 occurs in one cell. Okay, meiosis 1 occurs in one cell, and you're basically going to separate those homologous pairs from each other. When you're done with meiosis 1, you will have these two cells that you just formed. Meiosis 2 takes place in two cells. These two cells you formed from meiosis 1, and you're going to repeat all the stages again, and this time you'll separate the sister chromatids. So meiosis 1 separates the homologous pairs, and meiosis 2 that occurs in two cells separates the sister chromatids. And I believe I did an overview. Yes, I did. Let me stick I got to get smaller. Wish I was a bala. Okay, here we go. I don't know where to go. I'm in the middle. Okay, there you go. Sorry, delay of game. All right, so here we have interphase. We're starting the top left and we're winding all the way around. We're starting at my head and ending below. Okay, so here we have this cell, and it looks like it has 1, 2, 3, 4 chromosomes. Now, if it has 4 chromosomes, how many homologous pairs does it have? Right, 2 homologous pairs. Right, the 2 big ones and the 2 little ones here. And what's happening is the nuclear envelope is breaking down, just like what always occurs in a prophase, right? The chromosomes become visible, the nuclear envelope, the nucleolus breaks down, radiating microtubules, asters. That part is still the same. Now, there's some variation I'm going to talk to you about in just a minute, but for right now, let's just focus on that. And here in metaphase, you still have something in the middle, but this time, the homologous pairs are together in the middle, okay? And then in your anaphase, you're separating the homologous pairs from each other. Now, I want you to look at something here, okay? Do you see how the reds, lined up on different sides? They could have lined up together too, right? So they can line up, each homologous pair can line up how it wants to, as long as it's in the middle. And you have to pick a side, but it doesn't matter which side you're on as long as you're in the middle. That is called independent assortment, okay? Anaphase 1, you separate the homologous pairs. Here's telophase 1, accompanied by cytokinesis, division of the cytoplasm. We're reforming the nucleus. At this point, these two cells we formed in meiosis 1, they're considered haploid. Why? Because we have separated the homologous pairs from each other. Each of these two cells will undergo meiosis 2 in this diagram from right to left. Again, chromosomes becoming visible, nuclear envelope breaking down in prophase 2 now. Metaphase 2, notice how it looks very similar to mitosis, right? At this point, looks just like mitosis, but with half the number of chromosomes. Anaphase 2, the sister chromatids separate. Telophase 2, cytokinesis. So when we are done, we have a total of 4 haploid cells. Now, let me tell you where the kicker always is for people, okay? Listening up on this one, let me make myself a little bit bigger here. It's right over here. It's right there, okay? That's because both of these cells right here are both considered haploid. They're considered haploid because you have separated the homologous pairs from each other, okay? That's what makes them haploid. And then when you make these 4 cells here, you separated each of the sister chromatids from each other. So yes, it's still considered haploid. We haven't changed it, right? We just took a chromosome that consisted of 2 chromatids and we separated it just like we did in mitosis, right? But with half the number of chromosomes as before. Okay, so on your notes, 2 divisions. Meiosis 1, the top row up here, meiosis 1, you separate the homologous pairs of chromosomes, and you'll either receive mother's or father's chromosomes in each of these daughter cells. And then down here below is meiosis 2, occurs in each of the cells created from meiosis 1. Separate sister chromatids. And what are you forming right here? You're forming gametes. These could be 4 sperm that you made right here, gametes, which is a haploid sex cell. And then what's going to happen is these haploid sex cells are going to join together to form a diploid zygote. So look right here. We have 1, 2, 3 in this diagram. We have 1, 2, 3 chromosomes here, 1, 2, 3 chromosomes here. You reform your homologous pairs here in this zygote. Now we have a total of 1, 2, 3, 4, 5, 6, right? 6 chromosomes. So on your notes, zygote, a diploid somatic cell. Remember, a somatic cell is a body cell. A diploid somatic cell formed from the union of 2 gametes. And you have genetic recombination. You have a new combination of genes that's never been formed before, receiving a set of information from both the mother and the father. For us, it's over 70 trillion combinations with our 46 chromosomes and crossing over and all those options. So it's a new combination of genes never formed before, receiving a set, that goes in your notes, of information from both the mother and the father, over 70 trillion possible combinations. Now, non-sexual, we are sexual organisms, so we get this variety here. If you don't do meiosis, then what you have to rely on primarily to get variety is mutations. So non-sexual organisms have to rely on mutations for variations. All right, and let's take a look at the naked family here. I don't know why I can't have clothes on him. All right, so here is, I guess, dad. He is 2N diploid. Here is mom. She is also 2N. In her eggs and in her ovaries and in his testes, they will both undergo meiosis and form haploid sperm and egg. Now, it would be too difficult to draw, but there should be 23 chromosomes here and 23 chromosomes here. So these are the haploid sperm and egg. They are joined through fertilization to form the diploid zygote. Now, this zygote will undergo mitosis to form this ginormous baby. And then as this baby matures, it will continue to do mitosis. The only place they will do meiosis is this baby, whatever this baby is, a boy or a girl, either in its ovaries or its testes. All right. Okay, that is your overview. Let's see how you do here on my little test. Okay, so if I told you a 2N cell was 16, how many total chromosomes do you have? How many homologous pairs? How many chromosomes would you expect in a somatic cell? And how many chromosomes would you expect in a gamete? So you might want to hit pause, and then I'll go over the answers. Okay, ready? Okay, so if your 2N cell is 16, this right here, when it says 2N, that means it's a zygote, right? It means it's a somatic cell. It's a diploid cell. So if your 2N number is 16, how many chromosomes do you have? You have 16. Okay. How many homologous pairs do you have? If you have 16 total chromosomes and each of those pair up, then you have 8 homologous pairs, right? 8 chromosomes must have come from your mom and 8 chromosomes came from your dad. You have 8 homologous pairs. How many chromosomes would you expect in a somatic cell? Well, this is a somatic or body cell, so you would expect 16. How many chromosomes would you expect in a gamete? Gamete would have 8, right? It would have 8 chromosomes. 8 plus 8 would be then 16 in the developing zygote. All right, let's remind ourselves about homologous pairs. We have 23 homologous pairs. So they will undergo during the S stage of interphase. They're going to replicate. Meiosis 1 is about separating the mom and dad chromosome, the homologous pairs. And then meiosis 2, you would be separating these sister chromatids from each other. Okay, and here's an overview of that. So this is a cell that has four chromosomes total. So its 2N number is 4. It has a total of 4 chromosomes, 2 homologous pairs. And DNA is going to replicate. They're all going to double up. And in meiosis 1, little red and little blue are going to get separated from each other because they're a homologous pair, and big red and big blue are going to separate from each other because they're a homologous pair. Then these 2 cells will undergo meiosis 2, ending up forming 4 haploid cells, right? We started off with a cell with 4 chromosomes, and now it only has 2 chromosomes. So I want to go over the stages, which you already know, right? You've already learned all the stages. We just need to identify where the chromosomes are in our little dance here. All right, so quick overview of meiosis 1. So here's your nuclear envelope. Here's prophase 1. I know this is blurry. Don't worry about it. Here's prophase 1. You're going to have metaphase 1, anaphase 1, telophase 1, and cytokinesis, which will get you 2 cells. We're now considered haploid because we're down to just 2 chromosomes. Yes, those chromosomes are doubled up, but we still only have a total of 2 at this point. Remember, when the sister chromatids are together, they're considered 1 chromosome. And then here is meiosis 2 is going to happen in 2 cells, right? Each of those cells that we formed here, okay, Those two cells are each going to undergo meiosis 2. Look, same stages, metaphase 1, metaphase 2 now, anaphase 2 in both of these, telophase 2, and we have 4 haploid cells. Okay, so on your notes, you have stages, meiosis 1. In meiosis 1, prophase 1 is the same as regular prophase that we learned about in mitosis, except for this, okay? Homologous pairs attach through synapses. So do you see how they're all kind of snuggled in right here? And what's happening is when they snuggle in like that, they can, you have, let's say, chromosome 1 from mom and chromosome 1 from dad. When they synapses together, they can exchange part of the non-sister chromatids, and it can break off and exchange. And when that occurs, that is called crossing over. So there's some evidence right here of crossing over and down here of crossing over, right? Because we can see the exchange. When that occurs is during prophase 1. So on your notes, homologous pairs attach through synapsis, synapsis, through synapsis, and there's the word written out right there, and exchange portions of their chromatids, and this is called crossing over. This is called crossing over. Okay, metaphase 1, which you see happening here, you're meeting in the middle, but the homologous pairs, they can still be actually kind of connected together, and that's called a tetrad when they're all together. They line up here in the middle, so you line up with your dance partner. So metaphase 1, homologous pairs line up in the middle. Homologous pairs line up in the middle with either chromosome on either side, meaning that when they line up, it doesn't matter. You don't have to have all the dad chromosomes on one side and all the mom's chromosomes on another side. They can line up independently of each other. Okay. And then anaphase 1, you're separating the homologous pairs, so they're getting like pulled away, right? The homologous pairs are getting pulled away from each other. Homologous pairs separate. And then you're going to do telophase 1, where you're reforming the nuclear envelope here. And this is cytokinesis. This is an animal cell, so it's doing cleavage furrow. So telophase 1, same as before, 2 nuclei form, except may or may not be followed by cytokinesis. Now you're seeing complete cytokinesis here, but you may or may not do the cytokinesis completely. And I'll show you that at the end, why or why not. All right. You have all the notes for that. And then interkinesis is this between meiosis 1 and meiosis 2. It's kind of like the intermission between meiosis 1 and meiosis 2. Interkinesis, the resting stage between meiosis 1 and meiosis 2. These two cells right here, notice now they're considered haploid because they only have 2 chromosomes. A little blue, though it is doubled up, and a big red. Okay, so remember our original cell, let's go back here. Our original cell had 1, 2, 3, 4 chromosomes in it. Now our 2 daughter cells have only 2 chromosomes in them, so they're considered haploid. Right? Those 2 cells we just formed, they are going to undergo each one of them meiosis 2. Meiosis 2 occurs in 2 cells. So meiosis 2 occurs in the 2 cells generated by meiosis 1. All over again, you got prophase 2, same as before, except no crossing over. Why? There is no homologous pair to cross over with. And if it crosses over with itself, it doesn't really make any difference, right? So there's no crossing over in this prophase. Same as before, except no crossing over because there are no more homologous pairs. Metaphase 2, chromatids line up in the middle. Just line up here in this cell and that cell. Anaphase 2, sister chromatids separate from each other. Sister chromatids separate. And then telophase 2, right here, telophase 2, you're reforming those nuclear envelopes and cytokinesis, okay? 2 nuclei form in each cell followed by cytokinesis. So these could be 1, 2, 3, 4 sperm right here, right? Again, still haploid. All right, and in class, I'll quiz you a little bit on each of those stages and make sure you remember them all. All right, now, can this ever screw up? Yes, it can. And one way is through what's called non-disjunction, which is failure to separate. So normally homologous pairs should separate, right? And then sister chromatids should separate. Sometimes homologous pairs fail to separate and 2 go to one side. Or sometimes in this case, when you see non-disjunction right here, the homologous pair separated, and in one of the cells, the sister chromatid separated, but in the other cell, it did not. And so now this cell, these cells over here would be fine, but this one has one extra chromosome and this one has one less. Now, on non-disjunction, failure of either homologous pair or the sister chromatids to separate, and this can cause various syndromes. The most common autosomal non-disjunction is called trisomy 21, and most common autosomal trisomy, you would know this as Down syndrome. Typically short in stature, heart problems, intellectual disabilities, and chance of this syndrome increases with age of the mother. And this is why we're going to learn about that. Before, all of you that are biological females and have ovaries, you did meiosis 1 in all of your eggs before you were even born. You're like, check, done. Okay. Meiosis 2, where we separate the sister chromatids, that does not happen until a sperm comes knocking on our egg. And then we go, oh, okay, great. And then it separates. So the older you are, like I am, I always forget how old I am. I think I'm 53 or 54. I think I'm 54, okay? So my eggs and my ovaries are 54 years old. So some of the tiniest chromosomes have a hard time separating those sister chromatids because they've been stuck together for 54 years. So I would have a much greater chance of having a Down syndrome baby at my age than somebody who was 24 as opposed to being 54, right? 20, 34, and my eggs are 30 years older. The sister chromatids have been stuck together. That's why they highly recommend that you finish having your babies by the time you turn 40, because it really, the chances of having a Down syndrome child really increase after the ages of 40. All right, so now meiosis, the cool thing about meiosis is all the variability you get in the offspring. So that's the part I want to talk to you about next. So look at all these puppies, adorable. Here's dad, here's mom, and here are their 4 baby puppies. Notice none of them look exactly like mom and dad. There's a variety here, but they all came from the same parent. Why is there so much variety? So that's the part we want to go over. So on your notes, it says genetic recombination, variations due to the formation and union of 2 gametes made by meiosis. Variations due to the formation and union of 2 gametes made by meiosis makes sexual parents capable of adapting to changing environments. So let's see those 3 areas where we can see the variation, okay? First one, okay, remember when we did synapsis and we did the crossing over, and it's called a bivalent or a tetrad when it snaps together. That's going to increase variation. How much crossing over does occur and in which chromosomes? So on your notes, crossing over during prophase 1, that's the only time crossing over can occur, because that's when the homologous pairs are together and they do synapsis, and that's when crossing over occurs. So prophase 1, exchange of non-sister chromatids by homologous pairs. And I think I have in your notes exchange of sister chromatids. I'll make sure it says exchange of non-sister chromatids, or you can change that. Exchange of non-sister chromatids by homologous pairs. Okay, so that's variation 1. Oh, I got another picture for you. Up close and personal. Let's look at crossing over. There we go. So notice now all of these chromatids are completely different. This was, let's say if blue was dad from your dad, and this crossing over was occurring in your let's say ovaries. So this is completely dad. This is dad with a little bit of your mom, most of your mom with a little bit of your dad's genes, and then a gene from your mom, right? So all of these chromatids now are different. All right. And then the next way is in metaphase 1, when the homologous pairs line up in the middle, they can line up any which way they want. Remember, we have 46 chromosomes. Our 2n number is 46, right? So that means we have 23 homologous pairs. So all 23 of those line up independently of each other. So that is another form of where you could have variety in what gets separated to each of the gametes. If they lined up like this, then the gametes would either get all blues or all reds, right? If they lined up like this, then these would get the only variety would be in the smallest chromosome, right? So you can see all the different ways. And remember, we would have 23 of these all lined up here in the middle, 23 homologous pairs. So independent assortment, each parental homologous pair can line up either way during metaphase 1, either way during metaphase 1. And then the third reason we have so much, oh, I have yet another picture. Here's possibility 1. This is what you would get. Here's possibility 2. So we have 4 different options when you have these 2 homologous pairs. Okay, and then the third way is fertilization. You don't know what sperm is going to fertilize what egg. So fertilization, variation in which sperm will fertilize which egg. Which sperm will fertilize which egg. So now, comparing and contrasting mitosis with meiosis, let's look over here on the right on mitosis side. So here, 4 chromosomes, they double up. You learn this in video 1. They line up in the middle at metaphase, separate. You have a daughter cell that has the same number of chromosomes as the parental cell had. So this is a perfect clone. Over here in meiosis 1 is going to separate the homologous pairs. Then each of these cells is going to undergo meiosis 2. So on mitosis, number of divisions, you have just 1 division. It's right here. Whereas in meiosis, anaphase 1 is going to separate the homologous pairs, and anaphase 2 will separate the sister chromatids. How many daughter cells do you get? Well, in mitosis, how many did we get? 1, 2. 2 cells. Whereas in meiosis, we got 1, 2, 3, 4 cells. The results in mitosis, we have diploid identical cells, and in meiosis, haploid and not identical due to what? Crossing over and independent assortment. That's why they're not identical, crossing over and independent assortment. And how do we get genetic recombination in the baby? Well, then you throw in, you don't know which sperm is going to fertilize which egg. The location for mitosis is all tissues for growth and repair, and meiosis only occurs in the reproductive organs, And here's some charts. There are charts in your book. You feel free to pause on here if you wanted to screenshot those, if that would help you. Here's also a little Venn diagram I found on the internet for you, okay, comparing mitosis with meiosis, and this is showing you where they overlap. Okay, so that might be another helpful image to throw into your notes. All right, now let's talk about the difference between boys and girls. All right, so when boys do meiosis in their testes, right? So meiosis 1, you separate the homologous pairs, and then meiosis 2, you separate the sister chromatids. You make a lot of sperm, right? In the time that you've been watching this video, you probably made 20,000 sperm, okay? Now, girls are a little bit different. Okay, and this is why. Okay, the first division, yes, we separate the homologous pairs, but what happens is our cytokinesis is uneven, and this is why. Once the egg ovulates, right, it's going to take some time for the sperm to come and fertilize that egg. It's got to then travel down the rest of the oviduct into the uterus and embed somewhere in the uterus in order to get some nutrients again. And if you divide every cell evenly, then each egg then would have half as many nutrients and then half as many again. And so we go for quality over quantity. As females, we want to have as many nutrients as we can and increase the chances of any success. So we do uneven cytokinesis. The nuclear material is divided evenly, but the cytoplasm is not. And so when we do our second division, which happens once a sperm comes a knockin', we will undergo cytokinesis again unevenly. So we end up forming these large eggs, the ovum, and then these are called polar bodies, which will ultimately just degenerate. So nuclear division, what's going on here? Chromosome number, the same as males, but cytokinesis, totally uneven. Remember, males are they're going for quantity over quality. They're just trying to get there as fast as they can as members of the fallopian swim team so they can be the first one to fertilize that egg. And here is another picture showing you that same thing. Okay, so here's oogenesis, the formation of eggs, and here's spermatogenesis, the forming of sperm. So spermatogenesis occurs in the testes. Okay, so spermatogenesis, the beginning of sperm, right? Location is testes, and it results in 4 haploid sperm. 4 haploid sperm. That's one strategy, right? Quantity over quality. Oogenesis, different strategy. You're going for quality over quantity. So meiosis 1, right? Oh, and this occurs in the ovaries. Location initially in the ovary, but completes the process in the oviduct. Cytokinesis does not occur the second time until the sperm is there, right? So cytokinesis is done unevenly, to keep the nutrients in one large egg, So this part right here, girls, happens before you're ever born. Okay, then this sits in your ovary, it gets ovulated, and if a sperm shows up, then you will do cytokinesis 2, and then that sperm and egg can then fuse to form your diploid zygote. So cytokinesis is done unevenly to keep the nutrients in one egg cell, and what happens to these polar bodies that you have? Those polar bodies will deteriorate, just go away. All right, so that's it, my friends, and if you are in my class, I'll see you there.